Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science.
Please remember to like, subscribe, and share this deep dive with friends and family. It really supports the Science Experience.
When we talk about aging, the conversation usually goes to the big stuff. Joint pain, maybe losing some muscle, that sort of cognitive fog we all worry about.
Right, the visible symptoms.
Exactly. But if you really pull back the curtain on the biology of it, the problem often starts at the absolute smallest scale.
Yeah.
Inside our cellular energy factories.
That's it. We're talking about mitochondrial dysfunction. It's considered one of the main hallmarks of aging. And if you want to understand why things decline as we get older, you have to look at the power grid itself.
And today, we're zeroing in on the final, most important machine in that power grid.
We're looking at ATP synthase, or, complex 5 in the oxidative phosphorylation chain, what scientists call OXPHOS. And this thing is just this incredible multi-subunit rotary enzyme. It literally spins to churn out ATP, which is the energy currency for everything our cells do.
But the problem, and what's reason for this whole study, is that we've never really had a complete map of how this machine is doing across the whole body.
Exactly. We haven't had a high-resolution picture that looks at everything, considering both age and sex at the same time.
So the mission was born, create an atlas. A detailed, systems-level map of complex 5 activity. And they looked at 32 different tissues.
I mean, everything. Brain regions, parts of the GI tract, different kinds of muscle and fat. And they did this in young mice, 10 weeks old, and old mice at 80 weeks, looking at both males and females.
They wanted to see where the engine breaks down, where it just slows down, and kind of surprisingly, where it actually speeds up.
But before we get into that atlas, we have to appreciate the engine itself. Because complex 5 isn't just an ATP producer. The researchers point out something fascinating. It's actually a bidirectional nanomachine.
A bidirectional, so it can run forwards and backwards.
Precisely. When everything is going smoothly, it uses this thing called the proton motive force to crank out ATP.
But what happens when things go wrong? When the cell is stressed or maybe not getting enough fuel?
When that proton motive force collapses, the machine literally reverses. It stops making ATP and starts breaking it down, hydrolyzing it.
Wait, why would it do that? Why would it spend its own energy currency? That feels backwards.
It's the cell's emergency brake. By sacrificing a little ATP to pump protons back out, it helps restore the electrical gradient across the membrane. It preserves the mitochondrion and keeps the whole system from failing during, extreme energetic stress.
So its ability to run and reverse its hydrolytic capacity is actually a really sensitive marker for how healthy that mitochondrion is.
That's the key. And that's what they decided to measure. It's a brilliant way to gauge the enzyme's fitness at a massive scale.
Okay, but a massive scale. We're talking about 1,280 samples. How on earth do they do that consistently without spending decades just isolating mitochondria?
This is where the method is so clever. They didn't isolate them. They used tissue homogenates. Basically, they mashed up the tissue and measured the hydrolytic activity directly. They used a machine called the Seahorse XFEN96 analyzer. It measures the acidification rate. When ATP gets broken down, it releases protons, which makes the liquid more acidic. The Seahorse can detect that change with incredible precision.
So let's walk through that. You've got all these different tissues from muscle to fat. How do you make sure you're only measuring complex 5 running backwards?
They used a kind of chemical toolkit. First, to stop any forward motion in the chain, they added inhibitors for complex 1 and complex 3, rotenone and antimycin A.
So the forward assembly line is completely shut down.
Completely stalled. Then they add ATP and an uncoupler called FCCP. That combination basically forces the machine to run backward as fast as it possibly can.
Okay, so that gives them the maximum possible rate.
And then for the final check, the off switch, they add oligomycin, which specifically blocks complex 5.
And if the acidification rate drops to 0?
The activity you were measuring was from complex 5 and nothing else. It's a really robust system.
That's incredible. So with that confidence, what did this atlas actually show? Were energy demands pretty much the same everywhere?
Not even close. The first huge finding is just the striking tissue heterogeneity. Complex 5 activity is, it's all over the map, literally. It really reflects what each tissue is built to do.
So where were the engines running at full tilt?
No big surprise here. It was the contractile organs. the high-energy movers, the heart, of course, and big striated muscles like the quadriceps and the hamstring, the diaphragm, too.
And the tongue was right up there, wasn't it?
It was consistently one of the highest in young males, which you might not think about, but the tongue is for constant fine motor control, eating, talking. It has a huge metabolic cost.
That really brings the biology home.
Okay.
Okay, so on the flip side, which tissues were just kind of coasting along with low energy needs?
That list included parts of the GI tract, like the distal colon and the ileum, also the pancreas and the skin.
Tissues doing more secretion and absorption, not so much heavy lifting.
Exactly. And interestingly, gonadal white adipose tissue, GWAT, was also consistently near the bottom.
So this baseline map tells us that every organ starts from a different place. They have their own energy budget and I guess their own vulnerability to aging.
That's the perfect way to put it. Aging isn't this one global failure. We have different levels of resilience baked right into our systems.
Which sets up the billion-dollar question. What happens when you fast forward 70 weeks? From young to old, does the whole map just fade out?
It redraws itself, and it does so in really complex ways. The big headline here is that age has a much larger impact than sex. It accounts for the biggest changes, both in number and in magnitude.
Age is the dominant variable.
Without a doubt.
So the common wisdom is that everything just declines with age. Is that what they saw?
It's the general trend, yes, but not the whole story. We definitely see decline. In old males, for instance, there was a big drop in the tongue, the gastrocnemius muscle, and the kidney cortex.
And in females?
The decline was actually seen across more tissues, 7 of them, including the liver, spleen, skin, and really importantly, both major white adipose depots.
Okay, but here's where it gets wild. Some tissues didn't decline. They went the other way. Their activity went up with age.
This was one of the most surprising findings. The researchers think it might be a compensatory response. You know, maybe other parts of the mitochondria are failing, so complex 5 has to work overtime to keep the lights on.
So wait, is that a good thing or a bad thing? Is the machine running harder a sign of resilience? Or is it a sign of desperation before it burns out?
That is the critical question this atlas raises. It's hard to say for sure, but it shows a metabolic flexibility we might not have expected.
So which tissues were working harder in old age?
In old males, the list included the heart atrium, the diaphragm, and fascinatingly, that gonadal white adipose tissue, GWAT.
Whoa, hold on. You said in females, the fat depots crash metabolically. But in males, one of them is ramping up.
An enormous distinction. They're going in completely opposite directions.
And were there any tissues where activity went up in both sexes?
A very short list. Only 3. The diaphragm, the lung, and the quadriceps. Tissues absolutely critical for breathing and moving, suggesting they might be programmed to fight back against aging across the board.
So age sets the magnitude of the change, but it really seems like sex determines the direction of that change.
That's a great summary. The sex differences are highly specific to each tissue, and they evolve with age.
What did that look like in the young mice?
In the young mice, females had higher activity in places like the skin and pancreas, and much higher activity in their gonadal fat, the draught. Young males, on the other hand, had higher activity in the tongue and the cortex.
But the adipose tissue is where the story just becomes a perfect example of this divergence.
It's a textbook case. To recap, young females, much higher activity in GLOT than males, but with age, that activity in females plummets.
While in males, it skyrockets.
Exactly. They go in completely opposite metabolic directions, a total switch. The female fat depot seems to be shutting down, while the male one is trying to ramp up.
That feels like a massive clue for understanding how metabolic diseases, like diabetes, might progress differently in men and women as they get older.
It has to be. The researchers speculate it could be driven by changes in sex steroids over the lifespan. You know, the decline in estrogen in females might play a huge role.
That makes a lot of sense. The hormonal environment is tweaking the dials on aging. And this wasn't just in fat, right? How many tissues showed this kind of opposite direction change?
14 of them. Nearly half the tissues they looked at showed these highly divergent aging responses between males and females.
So we see these huge functional changes. Is it just because the cells are turning down the genes that make complex 5?
They looked at that. They compared their activity atlas to gene expression data for 19 related genes, and the key finding was only a partial match.
So it's not just a matter of the cell making less of the machinery.
Right. It strongly implies the changes are happening after the genes are read. It's what we call post-transcriptional mechanisms.
So what does that mean in simple terms?
It means the instruction manual might be fine, but something's going wrong on the factory floor. The proteins are being modified or they're not assembling correctly. It's a problem with the machinery itself, not the blueprint.
Okay, so with a study this big, there are always limitations. What were the big caveats we need to keep in mind?
They pointed out a few important ones. First, they only looked at 2 time points, young and old. We're missing the whole middle-age story.
The gradual progression.
Exactly. Second, the assay measures that maximal emergency reverse function. It's a great proxy, but it's not the same as the delicate balance of making and using ATP in a living cell.
And the fact that they had to mash up the tissues, that loses some context, right?
It does. By disrupting the mitochondria, you can't see changes in their structure, like the folding of the inner membrane, the cristae, and that structure is critical for how efficiently complex 5 works. Some of the decline could be structural, and this assay can't see that.
So to wrap this up, this deep dive really complicates that simple story of aging equals decline. We've learned that ATP synthase activity is totally different depending on the tissue. Age is the biggest driver of change, but sex dictates the tempo and, crucially, the trajectory of that change,
Leading to these wildly divergent aging paths, especially in places like fat tissue.
And the real value of this atlas.
The real value is that it gives researchers a baseline. When they study a disease, say Alzheimer's, and they see mitochondrial dysfunction, they can now compare it to this map.
So they can tell what's disease and what's just. The normal, messy process of aging differently for males and females.
Exactly. It lets them separate the pathology from the normal divergence.
Which is crucial. And it leaves us with a provocative thought. For those tissues that are in compensatory overdrive in old age, like the male fat tissue, should we be trying to slow that engine down? Or should we be finding ways to give it better fuel to help it run more efficiently?
That's the next big question.
We hope you enjoyed this deep dive. Remember to like and subscribe to keep up with all our newest material. And thank you for being a part of the Sand Science experience.